apparently it decided that it wanted to re-download the software. Shall we begin?
Oh, okay, hang on. I need to introduce you.
Oh, sorry. Yes, please.
Okay. Is New York in? Hey, Manji, are you there? Okay, she'll probably join later on. Anyway, I think we have Sheena from Kentucky, and then Melissa I can see, and obviously we have a group. We have, who's missing in our place? Okay, Hunter is not here, and then Tameen and John. And John here. Okay. Anyway, let's begin. I think it's a little past 3 o'clock, 2 or 3. So, it's my pleasure to introduce Dr. Aurijit Sarkar. He is a professor at University of High Point University in North Carolina. He was here with me. He spent in this building. Obviously, he spent probably 2 years. And 3.5. Oh, okay. All right. So he's a true drug discovery person. He's a computational person who does a whole lot of calculations and things of that sort. We have a number of papers together, and I think his focus is on infectious diseases, right? So I think this is one area where obviously glycans play a significant role, and I thought it would be good for him to lecture on this. Earlier I used to do these 2 lectures, but then he agreed to this. So he's been doing it for the last, 3 years now?
Something like that, yep.
So thanks, Aurijit, for taking care of this, and hope you have a good time with the setup that we have. I know you don't like online lecturing, but I would advise that all the centers, all the web, other than Aurijit, basically put on mute. And when you want to speak up, unmute and then speak. And I think generally we should hear from everybody. So today the setup is such that everybody can chime in by simply unmuting. Okay? Very good. Thanks, Aurijit.
No problem. Thank you, sir. So I hope everybody can hear me, and I'm really excited to be doing this lecture. Let me just begin by saying this is, anytime you have a question about anything, if I can help you in any way, please do chime up. Please just unmute. Let me know what your question is. But this is typically a very hands-on type of lecture. I like a lot of interaction. I do this in person typically. This is the first time I'm doing it online. I don't know how well this is going to go, but I'm going to give it my best shot. And under any circumstances, if you have any questions, please do email me at any point of time. My email address is right there at the bottom of this slide. Email me. I'll be more than happy to respond to your questions. Right, so that said, this lecture is typically meant for students who are starting up with graduate studies, so it will require just a little bit of prior knowledge. It is highly introductory in nature. It is virtually impossible to cover the glycobiology of bacterial and viral infections in 2 hours. So please rest assured this is at a very elementary level. And if you would like to talk to me about details, chances are I'll also have to read up, but I'm more than happy to interact with you in any shape or form. My expertise is in drug discovery. I love talking about new targets, especially in bacterial infections. I'm particularly interested in looking into antivirulence compounds, and you might see a little bit of that during this lecture. But if at any point of time you become interested in this area and would like to get in touch with me, please do email me. I would love to hear from you. Right, to begin with, let me just reiterate the objectives for this lecture. We are going to have a very brief introduction in how glycans will play a role in bacterial and viral infections. In essence, bacterial infections depend on glycans in a number of different ways, which includes the actual structural aspects of various bacterial cell components, interactions with human hosts, the pathogenesis of the disease, virulence, antibiotic resistance, viruses too. The entire life cycle of the virus depends on multiple glycoproteins. Antiviral treatments can also comprise of drugs which are grounded in glycans. And I just hope to give you a short excerpt of all of these different things to get you interested in glycans in microbiology. So just to begin, brief overview to microbes. They're present everywhere. They're always there on living tissues, non-living surfaces. You and I are actually covered in tons and tons of bacteria. There are probably more bacteria present in our body than the total number of cells in our body. A very small, very small fraction of them actually are pathogenic. But when they are pathogenic, the recognition of host cells, host tissues, the invasion of those tissues, the evasion of immune responses, and proliferation will all depend on glycans in one form or the other. And as a matter of fact, glycans are involved in all of these processes, and I'm including not just the sugar part, but the complete glycan in all of this. Let's take some classical examples of glycans in bacterial infections. So I'm going to touch base on some of the common therapies that we use against bacterial infections to begin with. So for example, let's take a look at peptidoglycan and beta-lactams. So if you take a look at peptidoglycan, this is also known as murine or muropeptide. It is an essential component of the cell wall. Typically what happens is there's this precursor right here, which basically forms the outer cell wall comprising of N-acyl muramic acid, which is shown right over here, which is connected to a number of amino acids such as alanine, D-glutamine. It also has mesodiaminopimelic acid, and then this D-ala moiety. So this entire glycan is in reality the target of an antibiotic called vancomycin. You might know about vancomycin. It is often called the drug of last choice against gram-positive bacteria such as Staphylococcus aureus. So if you have a MRSA infection, vancomycin is often given, which targets this part, this D-ala part of the peptidoglycan precursor, which stops the mesh-like framework which would normally have been formed. And without this mesh-like cell wall, the bacteria cannot survive. This is how vancomycin actually works. So one of the ways in which antibiotic resistance, for example, would work in the case of things like vancomycin-resistant Staphylococcus aureus, which is a few species of Staphylococcus aureus, would be to modify this D-ala moiety into D-ala-D-lac. Vancomycin can no longer bind to this moiety right here. So this part right here is being changed. Not the sugar part. The sugar part will come later. We'll see what role it plays. But in terms of this whole glycan, the D-ala part gets modified to D-ala-D-lac, and it no longer works. Now, this actually happens because of something known as the Van operon, which, as a matter of fact, has recently been acquired by Staphylococcus aureus from Enterococci in order to become resistant to vancomycin. Now, again, just to reiterate, this is the non-saccharide part of the full glycan. But again, like I said, I consider not just the sugar parts, but also the non-sugar parts of glycans should be important. And it's all part of glycobiology, in my humble opinion. So let's just take a look at not vancomycin, but a couple of different classes of beta-lactam antibiotics. So this is typically what happens to the peptidoglycan precursor when it is being synthesized into the cell wall. There are two different things that could happen. One is DD transpeptidases might act on it, which are a target of beta-lactams, in which case there's one of the D-ala parts which is retained, but two such units have now been cross-linked. Again, the non-glycan part of a full glycan, non-saccharide part of a full glycan, but an important target for beta-lactams. If you have DD carboxypeptidases and LD transpeptidases, these are again the targets of carbapenems, which are a certain type of antibiotic, which eliminate this alanine part right here, but again give you a cross-linked structure. So beta-lactams act on the DD transpeptidases. The LD transpeptidases, if these are involved, these are actually resistant to beta-lactams, with the exception of carbapenems. So if you think about it, there are some questions that we could ask ourselves, such as what therapies would you use for a bacterium that contains a point mutation in the DD transpeptidases in the active site? If you'd like to chime in in response to these questions, please feel free. I'll give you about 5 seconds to try out. You can take a look at this slide if you like. Okay, so let's say that we have these DD transpeptidases, and you have a point mutation over here, which is causing your typical beta-lactams not to function anymore. So what you would do, for example, is to try out carbapenems, carbapenems would be used. Yeah, no, carbapenems would work, but also you could go for vancomycin, which might actually stop the formation of this by binding the precursor itself. So as you can see, glycobiology actually plays an important part, not just in the basic science of infectious disease, but also in which antibiotic you would use for therapy. Right, the next question you might ask is, what therapies would you use for a bacterium that expresses a beta-lactamase, which is the typical way in which microbes or bacteria would resist a beta-lactam antibiotic? Again, the answer is probably going to be vancomycin. And as a matter of fact, the primary way in which you would target a Staphylococcus aureus infection is by using beta-lactams. However, in case you have something like a MRSA, which expresses a beta-lactamase, then you would immediately go for the next line of therapy, which would be vancomycin. So now that I've given you a bit of an example of how important glycobiology can be in the science of infections, I'd like to introduce you to a more current topic in infectious diseases, which is, so we've talked about Staphylococcus aureus, which is targeted using beta-lactams. If it is a MRSA, then it becomes resistant to beta-lactams, in which case you would use vancomycin. Now, we've also talked about VERSA, which is a special kind of vancomycin resistance where the D-ala moiety of the neuropeptide has been modified. But you know what? The VERSA type of infections are not that big a deal at this present moment, because I think only about 10 to 12 of them have been identified across the last couple of decades throughout the world. A bigger problem is an intermediate type of resistance called vancomycin intermediate resistant Staphylococcus aureus, or VISA. These are strains of Staphylococcus aureus which they have a different mechanism to resist vancomycin. They don't modify the D-ala moiety. They do something really clever. If you take a look at this slide right over here, you have your control slide, control strain, two control strains as a matter of fact. This is mu50, which is a vancomycin intermediate resistant Staphylococcus aureus. What you see is that the outer layers, the cell wall, has become much thicker compared to these areas right here in the control strain. So what's going on in reality is that not just that the peptidoglycan layer has become thicker, but which quite clearly blocks access of vancomycin to the actual target, D-ala, which lies far below the surface, but it also creates a number of unlinked D-ala chains right on the surface, right over here. So what happens is your vancomycin is not really able to deeper targets which might have actually had the bacterium, and hence it becomes resistant to vancomycin to the tune of about, say, 2 to 4 micrograms per ml, in which case it is known as a vancomycin intermediate resistant Staphylococcus aureus. Again, it's thickening the cell wall, which is a glycated product in itself. And clearly, glycobiology is important here as well. So let's say that you have a VISA strain which keeps out vancomycin. Can anybody venture a guess as to exactly which other antibiotics might be affected by this thicker cell wall? Let me give you a hint. So vancomycin is your secondary therapy, right? What's the primary therapy that we just discussed? Is everybody able to hear me?
They know the answer, but they don't want to say anything.
Okay, well, all right. So the primary therapy is beta-lactams of some sort. That would be affected as well. Because you have a thicker cell wall, you need a larger concentration of beta-lactams in order to have an effect. The second question I have for you is, what about VERSA? Do you think VERSA can be treated by, clearly VERSA cannot be treated by vancomycin, but how would you treat it? Okay, the answer is beta-lactams, because VERSA, as we discussed earlier, a couple of slides ago, let me see, right here, would modify this D-ala moiety. This has nothing to do with the target of beta-lactams, which basically function at the carboxypeptidases and particularly the transpeptidases, right? So what happens is, even if VERSA cannot be treated using vancomycin, you can still use beta-lactams and it is treatable. So let's take a look at now the sugar parts, the saccharide parts of the murine layer, of the peptidoglycan layer. So the sugars are actually pretty important. For example, Glucnac followed by the N-acylmuramic acid is a typical structure that is found in peptidoglycan. Now, if you notice in this structure here, you see it's marked as R2. This R2 can be either a acetyl group or a glycollyl group. If it is glycollyl, then it is a special category of bacteria known as mycobacteria. The thing about mycobacteria is because of this R2 group, this modified saccharide, it is actually resistant to lysozyme. And the fact that it is resistant to lysozyme actually stops our body from destroying it using immunoglobulin A, which would typically have combined with lysozyme as part of our natural secretions to kill other bacteria. Mycobacteria are resistant to this form of immunity. The other thing that you should know about the saccharide parts is that the NOD2 receptor is the pattern recognition receptor that recognizes peptidoglycan in mycobacteria. Now, just to clarify, personally, when I was a PhD student and even a postdoc, I did not know anything about pattern recognition, about the NOD2 receptor. So I'm just going to give you a little bit of a background into it. Those of you who already know, please do forgive me, but I knew my weaknesses, so I'm trying to help others overcome those. So one of the ways in which our innate immune system recognizes pathogens is by recognition of patterns which have been conserved in pathogens for eons. These are known as pathogen-associated molecular patterns, or PAMPs. And these PAMPs are recognized by specialized receptors coded by our germline, such as, for example, the NOD2 receptor. So the NOD2 receptor is actually the receptor which recognizes the peptidoglycan in mycobacteria and helps our immune system get rid of these bacteria. So let's get to the structure of bacteria real quick. We were talking about murine, about peptidoglycan and the cell wall, which really is part of one of the structures that exist on bacteria. So let's take a look at this mythical bacteria that people talk about. They talk about how the DNA is present in the cytosol, which is enclosed in membranes. Perhaps it might be two, depending on gram-positive or gram-negative. There's a cell wall, there's cilia, flagella, whatever it is, right? They imagine bacteria to be in this way. This is the way I was taught when I was in high school. I was taught when I was in my undergraduate education, all the way through until I hit my assistant professor position, when I started to look at the structure of bacteria a bit better, and I found that this is more of what you can expect in bacteria. The actual cells are pretty small, but there's a huge glycan layer to the outside. And this is a polysaccharide, actually. It's a polysaccharide layer, excuse me. And this is known as the capsular polysaccharide, and it is a very important component of bacteria. The outermost layer of bacterial cell basically helps the bacteria stay hydrated, so it will retain a good amount of water so that it prevents desiccation. Then it will mediate adhesion of the bacteria to various surfaces, including live tissue or just any surface. It will promote colonization, and it will be very important for virulence. And yet, most of the antibiotics that have been designed have completely ignored this really thick layer of polysaccharide which exists outside of the cells. If you can take a look at it, the volume of the polysaccharide itself is at least as much as the cell, if not more. So the capsule can be of many different types. Now, you have come across the capsule previously, I'm sure of it, but let me just remind you in case you've forgotten. There are different types of polysaccharides, capsules. They are classified typically into various groups depending on the exact composition of the polysaccharide, such as group 1A would be hexuronic acid with some sort of a neutral sugar. Group 1B would be hexuronic acid, one of the hexuronic acids with N-acetylglucosamine. Group 2 would be some sort of a hexuronic acid with KDO or sialic acid and a neutral or amino sugar. So the composition actually changes, and along with the composition, the nature of the bacterium and the abilities of the bacterium change by quite a bit. Some examples, of course, include hyaluronin, which is produced by certain types of bacteria. And we'll come up with this in just a little bit. Now, K5 would be 1,4, beta-1,4 connected glucuronic acid and glucnac. K1 would be something different. This is nu5AC, as a matter of fact, which is, if I remember correctly, it's 8 carbons. This decides the type of bacterial strain that you're dealing with. So this, in immunology terms, we call it the serotype of the bacterium. So for some of the drugs that you should really be aware of include Prevnar 7, 10, and 13, which are in essence capsular polysaccharides from pneumococci, which are used as vaccines. So once you vaccinate a person using these particular vaccines, it helps protect against either 7, 10, or 13 different serotypes of pneumococcus. So one of the reasons why these capsules exist is called molecular mimicry. For example, Neisseria meningitidis has 2 different types of capsules. You could have group C, where you have alpha-2,9 linkages, or group B, where you have alpha-2,8 linked polysialic acid. The thing to know about this is group B is non-immunogenic. So if it's alpha-2,8 linked polysialic acid, the immune system cannot recognize Neisseria. Whereas if it is group C, then it is immunogenic in nature. So it also helps evade, so clearly it helps you evade the immune system. Strep A actually produces hyaluronin, which is virtually identical to human hyaluronin. I wonder if I actually mentioned this. I did not, probably. Let me just tell you one more thing. Another example of where hyaluronin would be extremely important in infections would again come from Staph aureus, which is one of my favorite microorganisms. Some of you might be aware of an enzyme called hyaluronin lyase. It is expressed by certain strains of Staph aureus, which what the staph does is it produces this enzyme and secretes it to the outside during an infection, which chews down hyaluronin into saccharides, which can be utilized by the bacterium as food. Was there a question by any chance? I thought I heard a microphone come on.
Not from here.
Okay. So clearly, as described in these couple of examples, these bacterial polysaccharide coats can be extremely important in virulence because in this example, it helps avoid the immune system. In the staph example that I gave, where I said that human hyaluronin is being chewed up by a virulence factor, which is being produced by staph, actually helps produce sufficient nutrition for that staph to survive and replicate. The one thing that you should remember at all times is that the human immune response to bacterial capsule is lowest at the extremes of age. So if you are really young or if you are really old, then your likelihood of responding appropriately to an incoming pathogen which possesses a polysaccharide capsule is likely to be pretty low. Which is one of the examples where infections of the mouth and throat, ear, sinuses, etc., happen more often in the very young. Or if you have some sort of pneumonia, for example, you're really young or really old people are more likely to be susceptible, much of which is actually attributable directly to the bacterial capsule. Another thing about virulence genes, which are, well, the capsule is in essence a virulence gene, or it is a factor which is expressed because of a virulence gene, and this virulence gene may actually be passed on from bacterium to bacterium. This is also something that I think might be of interest to a lot of people by horizontal gene transfer. So let me show you something that was done a lot of years ago. This was actually one of the earlier parts of evidence of horizontal, of the presence of genetic material which can be inherited from bacterium to bacterium, which is the process is known as horizontal gene transfer. So the experiment that was done was the scientists took mice and okay, let me begin with the definition of rough colonies and smooth colonies down here. If you take a look at rough colonies, the reason why they're called rough is because there is no surface capsule present, so virtually any part of the surface of these bacteria that can be recognized by the immune system is recognizable, in comparison to the smooth colonies where you have a polysaccharide capsule present, which in essence helps the bacterium avoid immune recognition. So if you take these rough colonies of strep pneumonia, we're talking about Streptococcus pneumonia or pneumococcus, you take these rough colonies of strep and you inject them into a mouse. The mouse is likely to survive because the mouse's immune system can actually respond to these rough colonies. You do the same with smooth colonies of bacteria, and the mouse actually dies because the immune system is unable to respond to these bacteria. If you take these smooth colonies and you boil them, in other words, you kill them, and then you inject the same into the mouse survives again. Why? Because now there is no bacterium that can kill the mouse. But the capsule is still there. Now, if you kill these smooth colonies and you mix them with your rough colonies, allow incubation, and then you inject a mouse, now even the rough colonies are able to kill the mouse. Why? Because the gene which allowed capsular polysaccharide synthesis, biosynthesis, has now been taken up by the rough colonies, and the rough colonies are now expressing the capsule, which avoids immune response, which I thought was pretty interesting. I remember learning about this when I was in high school, and I thought, wow, that's a pretty cool thing. But I never connected that with polysaccharide because I didn't know really what a polysaccharide was for the most part, especially its importance in bacteria. So I always bring up this experiment as an example from which all of us can learn. So again, many different types of bacterial serotypes can exist. So the same species of bacteria can demonstrate different types of outer layers, including polysaccharide coatings. And these are known as serotypes, which really can have different types of properties, particularly because of the saccharide composition. There might be other biomolecules involved as well, but polysaccharide plays an important part in this. So 5 major serotypes of meningococcus exist. this one can actually cause meningitis and brain abscess. 6 different serotypes of Haemophilus influenzae exist, which can cause an acute exacerbation of chronic bronchitis. 9 different serotypes of Group B strep exist. More than 90 different serotypes of strep pneumoniae exist, which causes pneumonia, sepsis, and meningitis, all of them. This is actually one of the reasons why we're not able to protect against all sorts of pneumonia, all sorts of sepsis. Because even if you are able to help the body recognize certain strains of strep pneumonia, other types of strains can also cause the same symptoms and the same conditions. So if we were to have different bacteria that produce the same polysaccharide, would it be recognized by the immune system if vaccinated using something like Prevnar 13? So let's say that the Prevnar 13 polysaccharide is being expressed by different species of bacteria, and you vaccinate using Prevnar 13. Would it protect or would it not protect? What do you think?
It should.
It should, correct. It does, as a matter of fact. Then let's say that what happens if a different strain of pneumococcus produces a polysaccharide that is structurally different from Prevnar 13? Would Prevnar 13 protect against that strain of pneumococcus as well?
The protection would be lost.
Say that again, please.
The protection would be lost.
Correct. You would actually have a lower chance of protecting using that particular. Actually, you would have no chance of protection using Prevnar 13 because the antigen is completely different. So as you can see, the bacterial polysaccharide coating also has a very important part to play in disease and in health. Again, which links back to glycobiology. Okay, so the biosynthesis of capsular polysaccharide is something that we should look at because thus far we have been focusing on killing the bacterium using substances which are toxic to it, such as antibiotics, which is pretty cool. But we know that there are strains of bacterium which have been found in 30,000-year-old permafrost, for example, where genes for resistance against those antibiotics already existed. Humans are relatively new to this entire game of resistance, really, because bacteria and fungi have been duking it out since millions of years ago. So, and most of our drugs, antibiotics, come from fungi. So it's natural that genes that resist antibiotics already exist. So there are certain other avenues which I personally would like to explore as potential, that have the potential to provide treatments, just that they're difficult to work with. One of them is perhaps the blockage of the biosynthesis of capsular polysaccharide. So one of the things that you must know about capsular polysaccharide is that since the substances that form this capsule differ, the biosynthetic process also differs. The primers may vary, such as like you might have lipid A or LPS formation compared to, say, KDO-based conjugates. These are typically always introduced to the reducing end of capsular oligosaccharides. So if you have a primer right here, and this is your sugar, then this is where your new sugars will be introduced. However, again, bacterium to bacterium, these biosynthetic transferases might differ quite a bit. For example, Pasteurella synthesizes hyaluronin by adding sugars to the non-reducing end. whereas strep adds sugars to the reducing end. So they work on different ends. Clearly, the structure of the enzyme differs, which means you will be able to, for example, target Pastorella separately from strep. Also, Pastorella uses an enzyme that is completely unrelated to any vertebrate, not just strep. So you can actually get selectivity for Pastorella compared to humans, for example. So less side effects is what we're talking about. So I think I may have already answered this question in that last discussion. So how likely is it that a drug that targets Pastorella transferase will target strep? The answer, quite clearly, is pretty low, because clearly these enzymes are completely different from one another, which allows us to target Pastorella separately from strep. But the question you have to ask yourself is there any reason why you might want to invest in such a drug? Any guesses? Okay, so in order to get to the answer of this question, who knows how or what the most recent news is with regard to C. diff infections? Yeah, Clostridium difficile. What are they doing which is turning out to be clinically efficacious, as a matter of fact?
They recently changed it so you don't get metronidazole anymore.
I heard something, if I got it correctly, something to do with metronidazole?
Yeah, they recently suggested that you don't use it anymore.
That is not functional anymore, did you say?
They just recommended that you don't use metronidazole.
They don't use metronidazole. Yes, that is correct. But what is proving to be efficacious?
Oral bank.
Okay, let me give you the answer. Fecal matter. A fecal matter transplant. So basically what you're doing when you take fecal matter from one person and you introduce it into the gut of another is that you are introducing a ton of microbes. You are rebuilding the microbiome. If the microbiome of a person's intestine has been destroyed by some sort of an antibiotic, then you have to rebuild it in this manner, which stops the C. diff infection in place. So what if you were able to identify drugs which really killed only the pathogen necessary and maintained the rest of your microbiome? Wouldn't that be interesting? At least I think it would. So that's the kind of a situation where you might want to have antibiotics which are selective for certain pathogens that does not affect the rest of the microbiome. And clearly this is also very important. For example, if you do a urine analysis or you take any sort of bodily fluid, particularly from the GI tract or the pharynx, etc., and you do a culture, it's very common to come across multiple microorganisms that really are there because they are there. They're not really causing a disease. And there's only one or two particular microorganisms in there which are really causing a disease. So those and leave the rest, might be better off in killing everything, which is what the current broad spectrum antibiotics do. This is why this kind of research is really important. Again, pertains to glycobiology. Okay, so we're continuing with the discussion of bacterial serotypes and biosynthesis of capsular polysaccharides. Pastorella uses a unique biosynthetic enzyme with two glycosyl transferase domains. There's the aminoglycosyl transferase domain, and there's the glucuronal transferase domain. It is found in very few other microorganisms. For example, E. coli is Klebsiella, and there is no human equivalent for this particular enzyme. So quite clearly, if you were to inhibit this particular enzyme, either domain, you would find an antibiotic which stops the growth or at least colonization by Pastorella, by Klebsiella, but would not cause as many side effects as you would see in other kinds of antibiotics. Now we come to a really important topic, and you go through one particular case which I think you might find very interesting. I've always been fascinated with lipopolysaccharide. This is one of the components of the capsular polysaccharide, particularly in gram-negative bacteria. You don't get lipopolysaccharide in gram-positive, only in gram-negative. And it has all sorts of biological activity. The major reason for that is that it is highly immunogenic. If you have a large enough colony of gram-negatives and you give it, let's say, third or fourth generation cephalosporin, which is able to kill that gram-negative bacterium, what happens is, upon death, the gram-negative bacterium will release all of this lipopolysaccharide, which you see up here, to the outside, which will cause a very bad immune response, which may even result in septic shock. So where exactly is this in gram-negative bacteria? If you know anything about gram-negative bacteria, you know that it has two different membranes, right? So this is the lower membrane, this is the outer membrane, inner membrane and outer membrane, excuse me, when you have a thin peptidoglycan layer, and here, the outer layer of the outer membrane is your LPS. That's your lipid A, and then this is your saccharide part right here. So this is what the structure actually looks like. It's basically divided up into multiple zones. There's your lipid A zone. You can see these fatty acid chains also over here. Then you have your glucnac and your KDO, two different layers, followed by the inner core, outer core, O antigen. The O antigen is the part which is most immunogenic. So now, memorizing all of these different details of the composition is probably not going to be useful to you at this point, unless you really want to specialize in lipopolysaccharide. But I do hope that you will notice that most of this is glycan. Most of this is glycan. And for example, the core polysaccharide would have a composition, something like KDO, KDO, heptulose, heptulose, glucose, galactose, glucose, and glucnac. The KDO part is always invariable. If you can block the synthesis of KDO, it basically stops your lipopolysaccharide from forming. The rest of the core is variable, and clearly that's the reason why you see an adaptive immune response rather than an innate immune response. More than 20 different sugars can be present in the O antigen right here. So as long as you have gram-negative LPS in gram-negative bacteria, it will actually be resistant to multiple types of antibiotics, detergents, bile salts, mutagens, you name it, it's resistant because the LPS layer keeps everything out. Only select agents are able to penetrate via diffusion because of the presence of LPS. So it's more likely that antibiotics such as beta-lactams can get inside the bacteria, not by diffusion, but only by the use of structures known as porins, which allow water and other nutrients to flow inside. So these days it is actually understood that because of the presence of LPS, in order to really identify antibiotics which will get inside bacteria, which is critically important for antibiotic design, you must be able to design those molecules which will penetrate through porins. Lipid A assembly is essential, quite clearly, because if this entire outer layer is gone, then the bacterium is not really able to survive. Again, lipid A, while it is not the saccharide part of lipopolysaccharide, it is a glycan. So it is also known that if you change the composition of your O antigen, then small changes will actually cause large changes in pathogenicity. Your immune response modulation can change. Your ability to do molecular mimicry can change. Clearly, because the outer part of the O antigen is exactly where your body will interact with the bacterial surface. Lipid A is a heat-stable endotoxin, and immune responses are actually pretty strong against it. And as I had mentioned earlier, if you kill gram-negatives, the entire lipopolysaccharide is released into the bloodstream, and not only the O antigen but also lipid A can interact with your immune system, causing an immune reaction to it. So here's a very interesting case. At least I found it hilarious when I read it. This middle-aged laboratory worker was brought to the emergency department because he was feeling malaise, headaches, nausea, vomiting. The patient was awake but listless with a pulse of 114 per minute, a blood pressure of 42/20. 42/20 millimeters of mercury, and an oral temperature of 40 degrees Celsius. The patient was treated with intravenous fluids, and a dopamine infusion was started at a dose of 5 micrograms per kilogram per minute. Blood cultures were obtained, and vancomycin and gentamicin were administered intravenously. The results of urine analysis suggest I'm assuming this has something to do with an X-ray. I've never heard that word before, except in this paragraph. And electrocardiography were normal. The patient was admitted to the medical intensive care unit with a presumptive diagnosis of septic shock. I'm wondering, clearly this has something to do with LPS, but I'm wondering what you think might have happened. The answer is on the next slide, so please don't look. Try to guess.
Septic shock.
Well, yeah, of course. Clearly symptoms of septic shock. What do you think might have caused it? Okay, I know, it's a bit difficult. What happened was that the gentleman had recently been diagnosed with a tumor, and he heard from somewhere that LPS might actually induce your immune response to be stronger so that it would destroy the tumor. So he self-administered himself with 1 milligram of pure Salmonella Minnesota endotoxin. The dose was 4,000-fold higher than anything that had ever been tried in clinical trials. Luckily, the patient survived and the appropriate antibody was administered using passive imaging. So just something to remind you of when you think of LPS, endotoxin, things of that sort. All glycated products, many of them are immunogenic. And it's just super interesting to think of LPS in that manner. So speaking of lipopolysaccharide and septic shock, what exactly happens in the inflammatory response, of course, is the bacterium has LPS, which goes ahead and interacts with cell like receptor 4, with the CD14 co-receptor, which ends up activating IRAC and many of the other kinases, kappa B, which goes ahead and induces gene activation for pro-inflammatory mechanisms. So basically, I'm not expecting anybody to memorize any of these things. Just remember that LPS is an excellent activator of inflammation, and that the response can be pretty wild, systemic, and strong. Might actually end up killing you rather than helping you out. So let's just take a look at the modifications of LPS. Salmonella enterica, you have a 4-amino arabinose added to the phosphate of the LPS core, which reduces the negative charge which is present, which protects from positively charged antimicrobial peptides. So that's a really neat mechanism by which this particular Salmonella species can protect itself against many of our bodily natural defenses. Pseudomonas aeruginosa actually has a hexaacetylated lipid A connecting palmitate and 4-amino arabinose, which is more hydrophobic, which again repels antimicrobial peptides. Yersinia festis, which causes the bubonic plague, at 21 degrees Celsius versus 35 degrees Celsius, its LPS changes. So at 21 degrees Celsius, it produces more of hexaacetylated LPS, which is more immunogenic, whereas at 35 degrees Celsius, which would be more of the body temperature that it would encounter within the human body, then it starts producing tetraacetylated LPS, which reduces to acetyl groups, which is less immunogenic. So a really neat mechanism by which Yersinia is able to protect itself. Adhesion and colonization by bacteria also depends quite a bit on saccharides and glycans. Clearly, in order to have an infection of any sort, first, the bacteria must be able to stick to your body in some manner or the other. So bacteria actually produce something known as adhesives. These factors involve sticking to the host tissue or tissue surface. Some examples are lectins, which are glycan binding factors. Others are glycosaminoglycan binding factors. All of this basically helps decide the tissue tropism of exactly how microbes can selectively colonize certain tissues but not others. So some examples are fimbriae, and of course there's also afimbrial adhesion. So some types of glycosaminoglycan binding proteins might be heparan sulfate binding, chondroitin sulfate binding, dermatan sulfate binding. Each of these, there are differences between different factors. You can have lectins, which are different types again. C types, lectins, E types, I type, L type, etc. The important thing for you to remember is that, again, either the bacterium is producing a sugar which helps stick to human tissue, or else it is sticking to a glycoprotein expressed by the human being. So an example of where fimbriae, for example, might come in handy, also pili, is, like I said, in adhesion. So an example where this becomes really important is in the case of E. coli, which causes urinary tract infections. So we know for a fact that young boys and girls of all ages and older men are extremely prone to UTIs. The reason why young boys, infant boys, are particularly prone to UTI, as are women of all ages, is because E. coli, which is the major pathogen in urinary tract infections, actually express pili and fimbriae. And E. coli can travel from feces to the perineal region, followed by the urethra and all the way up to the bladder, from where it can continue to express these pili and fimbriae, which are again glycated structures, all the way up the ureter to reach the kidneys, where it can cause pyelonephritis, bladder infection that causes cystitis. All of this is made possible by the expression of pili and fimbriae. Although there are certain other things which I'm not describing in this figure, but They will express biofilm, which we will touch upon a bit later, which protects from, say, attack by neutrophils, so on and so forth. Afimbrial adhesion, an example of afimbrial adhesion would be Bordetella pertussis, which quite clearly causes pertussis or whooping cough, which uses sialic acid containing glycans on the host lung cell surface. So here's the host lung cell surface, which expresses certain glycoproteins, and these glycoproteins are being recognized by receptors on the surface of Bordetella pertussis. Again, glycans in addition. Biofilm is one of my favorite types of infection-enabling phenomena. So you see biofilm right there and right there in multiple types of imaging techniques. What you see is basically a mixed coat which comprises often, to a large extent, of polysaccharide. This polysaccharide is known as alginate. Fun fact, alginate is also a major component of ice cream. So I'm sorry if I've ruined ice cream for you, but really, same substance. The thing about biofilm is the way it is formulated. We think of biofilm as a layer which is basically protecting bacteria deep inside, which is not necessarily true. If you look at it carefully, it has gaps and openings everywhere. So here's a diagram which explains it. This is your alginate layer, which also has certain other components, but we'll stick with alginate for this general description. Here are your different types of bacteria present inside it, some of which are metabolically active. Deeper bacteria, which are well protected, are actually metabolically low, so they are well protected. You do need food and water while you're inside the biofilm. So there are channels inside this biofilm to enable nutrients and water to get in to a certain extent, which keeps the bacteria alive. This, of course, is an example on tooth enamel, but this happens also on surfaces such as catheters, which are probably one of the biggest reasons why we have nosocomial infections these days. Anytime you go for a surgery, you have a catheter inserted inside you. If it has not been cleaned properly, or if for some other reason the bacterium gets in, Pseudomonas is really bad about this. Staphylococcus epidermidis is another one. They're both biofilm forming. They will end up forming a biofilm and cause a lot of trouble in your treatment. So as you can see, bacteria use glycans and glycoproteins and other glycated structures extensively in multiple functions, including invasion, adhesion, virulence, damage to tissues. Everything involves glycobiology in some manner or the other, which is the reason why I'm really interested in studying glycans and glycosyl-transferases in bacteria. I haven't talked about any of my work yet because I'm still gathering preliminary data. Perhaps in future versions of this talk I might discuss such things in a lot more detail. But let's, can we do a 5-minute break? I'm sorry, I think we probably should. That's my throat's kind of hurting. I need some water.
Yeah, let's take a 5 minute.
Yeah, see you in 5 minutes. Thank you. Right, I'm going to begin. If there's anybody still missing, please just speak up and let me know, okay?
All right.
All right, so glycans and viral infections. So we've talked about bacteria, viruses. The discussion is going to be much simpler and much shorter because, truth be told, while glycoproteins are extremely important in viruses just as much as bacteria, our successes in drug discovery against viruses are, excuse me, are lacking to quite some extent. So there's only so much we really have that we can discuss in the case of viral infections in terms of glycans. So, excuse me. So, let me see now. Okay, there we go. So the influenza virus, of course, being the primary virus that we can discuss. Influenza has a negative stranded RNA inside it for genetic material. It encodes for exactly 11 viral proteins, of which everybody must be familiar with hemagglutinin and neuraminidase. Hemagglutinin is responsible for recognition of the host cell, and neuraminidase is responsible for release of new virions which have been produced by the infected cell from the infected cell surface, which enables the virus to spread to fresh tissues and continue its infection. So there you see a typical example of the structure of a virus. There's your genetic material. These are your outside proteins. Some of them are structural proteins, such as M1 and M2. Neuraminidase and hemagglutinin, of course, are on the outside. There are a number of other proteins as well. Most, if not all of them, are glycoproteins. It's interesting because of how involved glycans are in the entire process of viral infections and spread of infections throughout tissues. So let me just go by the slides, of course, because I tend to start talking, giving out all of the information right at the very beginning when I still have slides left towards the end. So let me just follow the slides real quick. There are 4 different types of influenzas, A, B, C, and D. Only type A and B actually infect humans, of which only type A has caused pandemics so far. So if you take a look at the virus, like I said, neuraminidase helps release the virions from infected cells. M2 is an ion channel, which is again a glycoprotein, which is an antiviral target. Hemagglutinin binds to sialic acids on the cell surface, which is a vaccine target. And also, I should mention that neuraminidase is the target for oseltamivir and zanamivir, which I think we will discuss towards the end, but they are glycan-based drugs. And typically for each virus, you will have a certain description, which I really should cover. This is the type of description that you will see. This first part, A, is the virus type, so it's influenza A. Fujian is the geographic origin where it was first found. This will give you the strain number. This is the year of isolation of the strain, and followed by a subtype where you describe the hemagglutinin and neuraminidase types. That helps you understand exactly how the virus will function. So some of the things that you must know about hemagglutinin and neuraminidase are that depending on the type, you will have a different type of virus. The hemagglutinin, like I said, recognizes sialic acids on the cell surface for entry. But there are multiple types, 16 different subtypes as a matter of fact, H1 through H16, and neuraminidase, which cleaves sialic acid from the surface of infected cells to release the virions, also shows 9 different subtypes. Depending on which hemagglutinin and which neuraminidase you have combined together to form a virion, you may have different designations such as H1N1, H7N9, so on and so forth. Now, one of the things that you should know, for example, is that pigs are known as a mixing bowl for human and avian flu, because the human influenza virus, humans contain alpha-2,6 linked sialic acid, which is recognized by HA with a leucine at position 226, whereas birds have alpha-2,3 linked sialic acid, which is recognized by HA or hemagglutinin, which possesses a glutamine at position 226. Pig has both alpha-2,3 and alpha-2,6 linked sialic acid, which allows the human strains and the bird strains to mix in pigs producing strains which will allow cross-infectivity of humans with avian flu. Now, clearly, the avian bird flu has been a massive issue across the last couple of decades across the entire globe. I believe I believe even within the last couple of years, there have been instances where, especially in China, where they had to put down entire batches of birds because they wanted to kill off the bird flu virus before it could spread to humans. This is one of the reasons why we must know about glycoproteins in influenza, and why it's important overall. Now, clearly, the viral life cycle plays an important part in how we target viral infections. So, just a quick overview for those of you who may have forgotten or might not have heard of exactly how this works. Hemagglutinin, in the case of influenza, would recognize receptors, in this case sialic acid containing receptors on the cell surface, which allows adhesion. Then the virus inserts itself inside the host cell and releases its genetic material, which then takes over the genetic material of the host cell by inserting itself or its respective cDNA inside the host cell's DNA, which then takes over the protein production machinery, which allows for multiple copies of not just the genetic material, but also the necessary proteins to be produced by the host cell, which then buds off in the form of a virus eventually, where neuraminidase, cleaves off any sialic acid which would have caused the new virion to stick to the infected cell and spreads to nearby cells. So viral entry, hemagglutinin, requires saccharides, sialic acid. Viral release also requires cleavage of saccharides. Saccharides clearly are very important. Now, if you were to target something like neuraminidase, now this is an actual example that I've taken out of literature. So just to make you aware, this would be a plausible strategy where you could perhaps design, use structure-based design in order to identify novel inhibitors based on the mechanism of this entire process. Let's see, what do I have here? Okay, so basically this is what happens. This is your saccharide which is being cleaved by your neuraminidase. If you're a big fan of enzyme kinetics, you will know that such mechanisms actually allow for a transition state to be formed. The transition state can be mimicked in multiple ways, such as by these inhibitors over here, zanamivir and oseltamivir, which have really good affinities for neuraminidase. Once you inhibit neuraminidase, the new virions are just unable to spread to newer cells, fresh cells which have not yet been infected, thereby the infection is contained to very few cells. If you know your immunology, these infected cells are eventually destroyed by the lack of major histocompatibility complex type 1, and so the viral infection can be stopped from spreading. Again, we also would need to talk about, since we're talking about drug discovery, if you look at oseltamivir and zanamivir, oseltamivir is also known as Tamiflu. It's actually available over the counter these days. Zanamivir is actually a much stronger binder of neuraminidase. The thing with zanamivir is it has this guanidino group, which is the reason why it's nanomolar in its affinity, and yet at the same time, it cannot be given orally. So oseltamivir or Tamiflu you take orally, but zanamivir cannot be given orally. And the reason for that is because of this guanidino group, which is highly charged, so it's not orally bioavailable. But it can be inhaled, and it's very effective in that manner. So people did go ahead and design some mechanism-based inhibitors. If you look at the interactions, we can literally try to mimic these same interactions and try to identify inhibitors that would work in a manner similar to zanamivir and oseltamivir. This is for influenza. Now, actually, I guess I must have, I'm sorry, I'm just going to speed through real quick to get to this end. Oh yeah, okay, I put it in at the end. I'll get back to it eventually. So, another example of a virus that utilizes glycans in order to identify the cells that it infects would be the herpes simplex virus. It consists of a family of over 100 different viruses. The worst ones, of course, are herpes simplex, which end up causing keratitis of the eyes or genital lesions, which is sexually transmitted. Probably another pathogenic one, which is much less terrible, would be the Epstein-Barr virus. So there are a ton of different types of viruses that belong to this particular family. The thing is, the thing with these viruses is, for example, that it remains latent in the neurons for years and can become reactivated by even environmental triggers such as stress. So once you've got it, chances that you will see it again are actually pretty high. And it can also be contagious when the person who has that infection is still asymptomatic. Nearly 80% of the human population actually carries HSV1 compared to HSV2, which is still present in about 40% of humans. HSV2, of course, is the sexually transmitted version, which causes genital lesions. Again, glycans are extremely important for everything that the virus does. For example, what it does is it recognizes heparan sulfate proteoglycan on the host cell surface. Then it basically goes through a process called surfing till it recognizes a co-receptor, which is the GD receptor, which helps the virus move into the host cell surface. Then it goes through the typical process where it inserts its genetic material into the nucleus, takes over the cell, helps replicate itself and release itself again from the host cell. But again, the beginning part was heparan sulfate, which is a glycosaminoglycan. So again, there are multiple proteins which are involved on the virus's part, which are almost all glycoproteins. So both the attachment of the virus to the host cell are based off of glycoproteins. What was I trying to say here? Okay, so not only are the viral proteins glycated, but also the glycosaminoglycan on the host cell surface is, of course, clearly a glycan. In other words, again, glycobiology plays a really important part in how the herpes simplex virus gets inside host cells. So it has been demonstrated previously that the sulfate at the 3 position is extremely necessary for this virus to get inside the host cell, for recognition of host cells. So the structure of the glycan is just as important as the presence of the overall glycan and negative charge. So again, the reason why I put up this slide is to just make everybody aware that glycosaminoglycans are, I'm sure everybody knows this already, but just to reiterate, glycosaminoglycans are not just a source of negative charge. The microstructure, the functional groups which are attached strategically at certain positions will help or hinder many different biological processes. So it's actually a stigma attached with glycosaminoglycan research, is that everybody looks at it as just pure negative charge. That is not true. That's something that we need to give up. We know that is the case in blood coagulation. Here's an example in infections as well. We know there are several other situations where the exact structure of the glycosaminoglycan plays a very important part. Okay, so I'll just give you a bit of a background into my own research, which was on the human para-influenza virus type 3. This part of the research has actually been published, so here's the citation. You can just look at it if you want the finer details, but I just wanted to introduce everybody to a really exciting prospect that I think we found something interesting and then we never followed up on. So eventually, hopefully, things will change and we can focus on this project a bit more in the future. If not me, then at least the people who originated this project should look into it. So this requires an overview of exactly how the human para-influenza virus type 3, or HPIV3, functions. So it has a protein called hemagglutinin neuraminidase, which, in the case of the influenza virus, we had two different proteins. We had hemagglutinin and we had neuraminidase. In the case of HPIV3, it has one protein with two different functions. It has both the hemagglutinin function and the neuraminidase function. Hemagglutinin basically recognizes the sialic, or HN actually recognizes the sialic acid present on host cell surfaces, and then by an unknown mechanism triggers the fusion protein, which is again another glycoprotein, to trigger. That's the best description I can give you. It's called the spring-loaded mechanism. So what happens is, upon recognizing a sialic acid, this fusion protein shoots out like a spring-loaded mechanism, like almost like a gun. It inserts itself into the host cell surface, and then it changes conformation. It folds over, which causes the virus to come in and fuse itself with the host cell surface. So one of the ideas that we were exploring at that time was, what if we made the virus think that it had detected sialic acid using a drug-like molecule? Can we make the virus actually automatically trigger earlier than necessary? Would this stop the virus from replicating? And the answer was yes. As a matter of fact, it is possible. So we found three different molecules which, when these molecules bound the correct site, not site 1, but site 2. When they bound site 2, HN, hemagglutinin neuraminidase, would literally think that it is in contact with sialic acid. And it would automatically trigger the fusion protein, and the virus would become completely inactivated. That was a really exciting project that I was a part of. We did discover a bunch of other molecules which are distinct from these three, which would block the recognition of sialic acid at site 1 competitively as well. But that's a different story for a different time, I think. And as expected, I think I finished the lecture about 30 minutes too early. If you have any questions that I can help you with, I'm available. Please ask away, or else you do have my email. So please don't hesitate to email me at any given time.
Cool. Thanks.
No problem.
All right. Let me check with Melissa. Is she there? Okay, Sheena, Melissa. Any questions? There's Melissa.
Hi, we're good here. Thank you, Aurijit.
Okay.
Cool.
All right. Thanks, Aurijit. That was
No
great.
problem.
I think it worked out well.